My name is Jenna Dunn. I'm the Admissions Outreach Coordinator here at Keck Graduate Institute. We are very pleased to have you here with us today, and we really thank you for your time to come here today and listen to our wonderful professor speak a little bit more about his expertise and give you a little bit more information about what he knows and what you could learn potentially if you come to Keck Graduate Institute. So I would like to introduce and turn it over to our professor, Dr. Animesh Ray.
Thank you. Hi, everyone. I don't think I'm seeing everyone. How many of the students are there? I mean, candidates?
There
Five,
are
four.
3 right now. Yes, there.
Okay. And some are coming in as we speak. All right. Let's see. Yes. Okay. Very good. All right. Thanks for the introduction, and let me begin. I have a slideshow, probably already too many slides, but let me go through it and stop me anytime that you wish, okay? All right. And just unmute yourself and say, may I ask a question or something like that. All right. Okay, let me share my screen. All right, can you see my screen?
Yes, we can.
All right. Let me make this screen a little small so I can see, but somehow my cursor is not looking. Just one moment. All right. So AI to predict antibodies, and that's the problem. So if you think about COVID-19, and we are out of the pandemic officially, but the rest of the world are not. As of yesterday, the total cases in COVID-19 had been 627, more than 627 million globally. And that's an under counting because there are many, many COVID-19 patients who had actually died of this pandemic, but nobody knows that they had died of COVID-19 pandemic because in less fortunate places in the world, there are not that many diagnostic laboratories to look at every case. But so far as we know, there are at least 627 million cases, and 6.5 million, more than that, total deaths out of COVID-19. But since the vaccines were approved, there have been a lot more people that have been vaccinated. That's 12.8 billion people have been vaccinated globally. So this has been a tremendous, the exciting time for medicine to be able to develop a vaccine so rapidly, unprecedented speed, and then be able to vaccinate 12.8 billion, roughly 13 billion people throughout the world within such a short time. It's just a remarkable achievement, and politics and business practices and medical sciences have come together to make this possible. But let me mention that the pandemic is still very much on in many parts of the country, including the United States. Within the last 28 days, there have been about 12,500 new cases, and about, and of course, we have been vaccinating also quite a large number of people. So that's not the end. We have now been looking at the rise of the Ebola pandemic in Uganda over the past 1 month. And the cases have been rising. Within the past few days, the cases have been falling, but that's probably only a statistical fluke. And most likely, this is going to be another pandemic, hopefully localized. But that hope may not be realized because a few years ago, uh, Ebola was a pandemic in Central Africa, which had claimed over 11,000 lives. And Ebola is a much more lethal or virulent virus than COVID-19 is, SARS-CoV-2. So we think that many such pandemics will arise in the future. And the question is, how fast can we get to the people with therapy, vaccine, etc.? And that's the idea. So how can you rapidly respond to an emerging pandemic? So first is to isolate the people who have been infected, right? That's the public health challenge. Quarantine them. And as you know from your experience in the United States over the last few years, it is not always politically appealing to be able to quarantine people, depending on the country. United States and Brazil have been rather difficult, but many other countries that could be achieved much more readily, although with tremendous economic and social costs. But that minimizes human cost. But then, once that is going on, when the public health policies are in effect, then the medical science comes in and hopefully develops some sort of an immunization strategy or therapy. And one of the first that we are able to develop is the so-called passive immunization, in which we can generate a serum containing polyclonal antibodies against the virus from a person who was previously infected with a virus but survived. Or we can possibly generate a monoclonal antibody, a single antibody that could neutralize the virus. So that's an example of passive immunization. And then active immunization with a vaccine, for example, the RNA vaccine or adenovirus vaccine that you have already been vaccinated with, hopefully. And ultimately, hopefully, an antiviral drug. And an antiviral drug development is slower. And for a novel virus, it may not exist. It may not be done ever. For example, we are still struggling with an HIV/AIDS virus. There is currently a good maintenance therapy, a multi-drug cocktail for HIV/AIDS, but really no single magic bullet. So it's the passive immunization with antibody that's the key to the rapid response. And we see that in the timeline of COVID-19 response as well. COVID-19 was discovered in November of 2019 by a group of Chinese physicians. And the structure of the causal virus, so-called SARS-CoV-2 virus, was identified, and then its molecular detail of its surface features at atomic resolution, near atomic resolution, was done and reported by February 19 of 2022. That's wrong, 2020. And the genomic sequence was done by 2020 as well, and remarkably, the FDA approval for the monoclonal antibody was made by the November of 2020. And in order to get to the monoclonal antibody, one needed the genomic sequence, one needed the cryo-EM structure where the antibodies can bind, and so on. This is a remarkable progress, of course, because generally, from the discovery of the causative agent, a virus, to getting the genomic sequence or the molecular detail of the virus's structural features on the surface, usually would take years. And a monoclonal antibody therapy would take years, and the RNA vaccine is new. By December of, again, sorry, it's not 2020, but December of 2020. 2020, not 2022. So this was an unprecedented speed with which this was done. There's no parallel in the past to this speed of accomplishment. And yet, by that time in December 2020, millions of people had died of the virus. So can we do it faster? So what are the challenges? Well, first, you have to get the genomic sequence of the virus, and from there, you have to generate fragments of the antigenic proteins. In this particular case, for example, the spike protein of the virus, which you're familiar with, no doubt. And then one can do laboratory techniques in which those little pieces of the spike protein can generate antibody-like molecules, which can be tested very speedily in laboratories for identifying some realistic candidates that might serve as a passive vaccine. And once they identify it, you could generate the synthetic monoclonal antibodies based on the winners of such efforts. And then ultimately you have to test in model animals and also in vitro in the test tube for learning about the efficacy of these candidates in terms of whether these antibodies can neutralize the virus, and also presumably some side effects that it might have. Then ultimately pick the best candidate and do human clinical trial. And we have seen Pfizer, for example, started the clinical trial first, and then other companies came up with an RNA-based vaccine, and within 3 months, the clinical trial was complete. But these second through the fifth bullet points, they take a long time to get to the clinical trial stage. And our effort in the laboratory is concerned with some of these steps. And very specifically, the genomic sequence of the virus can generate fragments of the putative antigenic proteins. This is a part that we are actively working on, and then also the in vitro techniques to generate the potential antibody-like molecules. And I will describe some of these. So here is what the spike protein, can you see my cursor? I hope you can see my cursor.
Yes.
This is this, yes, okay. So this is the spike protein's molecular structure. These ribbons and ribbon-like things and arrows, they are parts of the proteins that fold in three-dimensional structures, and these squares are little pieces of sugars that are attached to the spike protein surface. Now, there is a term called epitope. Epitopes are parts of the antigen. So this is the antigen that is a foreign body once the virus infects a host. The antigens are not continuous stretches of the protein. In other words, they're not so-called linear. They're non-linear. In other words, they fold in complex ways in 3 dimensions. And as a result, the body sees various parts of these proteins in a complex manner exposed on the surface. And it is only those surface exposed fragments of the proteins that become effective antigens. And you have to be able to recognize those. So it's difficult to tell which amino acid sequence will make a good antigen without a 3-dimensional structure. And until recently, there was no reasonable way to predict these novel antibody protein sequences, which are called epitopes, without knowing, which are called paratopes, unlike epitopes, which are part of the antigens. So the paratopes are those that bind to the epitopes of the antigens. So paratopes are part of the antibody. Until recently, there was no way by which you could predict what the antibody's paratope is going to be that would specifically bind to the epitope. And even now, there is no known way by which you can look at the 3-dimensional structure of the protein and directly go from the structure of the antigenic protein to predicting a novel antibody. There's no way that you could do that. And the reason that you cannot do that is fundamental. And that fundamental reason is the so-called central dogma of molecular biology. It's called a dogma because nobody has really found a way out of this yet, but there is no a priori way by which this is impossible. It is possible, but we think it doesn't occur in nature. So what is it? That information flows only from the nucleic acids to the protein and not from protein to the nucleic acid. So information flows between two molecules of DNA through replication, as you see here, double-headed arrow. Replication transfers the information from one double-stranded DNA molecule into multiple double-stranded DNA molecules. Or by transcription, in which the information that is encoded in the DNA can be transferred into the RNA structure and sequence. And then it is decoded in the formation of protein sequences, which are amino acids arranged linearly, which fold in complex three-dimensional shapes. And it is the three-dimensional shape that determines the protein's function. And function is like information. So, for example, I am a professor, so my function is to profess, and I profess information. So, similarly, the protein, let's say it is an enzyme, and it has a function that is to catalyze a chemical reaction, for example. And that happens because the amino acid sequences of the protein folds in a complex manner. And there is so far, no conceivable manner by which, if you give a protein structure and not its sequence, there's no way we can flow back that structural information into RNA or into DNA. Of course, if we give the sequence of that protein, we can easily do that. But there is no way by which simply the shape of the protein can be moved back into encoding the information that it contains in terms of its shape into the sequence of the nucleic acids. And that's because it's going to violate the central dogma of molecular biology. But it's a dogma. It's a dogma because we think that it is very difficult to violate it. However, the adaptive immune system does it all the time through evolution. So on the left here, there is a diagram that I stole from Khan Academy, where it shows in a cartoon form what happens when a human body, for example, sees fragments of an antigen. It displays the fragments of the antigen on the surface of a T cell, of a kind of lymphocyte in the human blood, and B cells talk to the T cells, and if the B cells talk to the T cells, and the B cells have a genetic rearrangement, which is like mutagenesis and recombination, in which variants are created all the time in B cells, one of these variants produces a protein that is very similar to the protein fragments that are displayed on the T cells. Then it begins to proliferate. But if it doesn't, then it dies. In other words, it's like evolution and natural selection. And in the middle, that's it explains that's what happens in a natural selection, in evolution. Random genetic variation happens, in this particular case, in the antibody coding genes. And then there is a selection for survival. If there is no accidental binding between the antigen and the antibody is displayed on the cell surface, antigen is outside, then it dies. The cell dies. And only cells that survive are those that have some affinity to the antigen. And those cells which survive better bind tighter, more tightly than others. In other words, the survival of the fittest in natural selection occurs within the human body, and ultimately a cell with a monoclonal antibody gene that encodes the highest affinity binder to the antigen survives and proliferates. And of course, many such cells proliferate within an infected patient or a vaccinated patient. So on the right-hand side, I show the approaches that today's scientists take in order to generate synthetic antibody molecules, and specifically the way we are trying to do it. So we generate the antigen-antibody interaction candidate structures within a computer. And when we somehow learn to represent the structural features for the real interactions between antigen and antibody, the antigen in this case comes from the virus, generate fake structures, and then play a game in which the fake structures foil a score for affinity binding that the real structures have. And then this is if the fake structures win, then there is a loss in the affinity, and we call it a loss function. So the approach is to minimize the maximum loss function and keep whatever remains, and then test the winning structure, which is basically a kind of evolutionary game played in the computer, starting from the three-dimensional structure of the antigens and the antibodies. So our strategy is that we start with many known antigen-antibody three-dimensional structures, and some of them we don't know, we just have sequences, so we use deep learning computer networks, which are called AlphaFold, made by Google's deep learning machines. So we make these computational structures as well as take real structures, and then do something called representational learning, in which we try to represent these 3D features in certain abstract manner. And some of these features use heavy computational techniques such as molecular docking and molecular dynamic simulation. Molecular docking is, you can think of fitting a lock to a key in the computer by rotating the lock around the key or key around the lock in all different conformations nearby so that something fits accidentally. And if it doesn't fit, then the score is very low. And if it fits, the score is very high. And similarly, molecular dynamic simulation represents little movement of large molecules of antigens and proteins nearby at a particular temperature that we define into the computer, and it jiggles around these structures and then sees whether the energy of interaction is minimized, and when it is minimized, it's a fit, and we try to score that energy minimization function in terms of how much tightly the two molecules fit. And from these data, we produce initial models of structures, and then we generate random fake structures to compete against the predicted structures. And then we play this minimax game, which is something like a tic-tac-toe game in which two players, in this particular case the real structures and the fake structures, try to fool each other and minimizes the maximum error between the binding. And ultimately that converges to a novel structure, not one, but several novel structures that win. And this is also very similar to the evolutionary game between the prey and the predator, which try to fool each other. And the preys try to eat the maximum number of predators with the maximum efficiency, minimizing its energy resources, and the predators try to fool the prey as much as possible by also minimizing the energy. So deeply, these computer games are very similar in mathematical structure to the prey-predator game or the evolutionary game that evolution plays on real subjects. And in this particular case, we make a specific type of computational model, and they're called generative model or discriminative model. What are they? So most models are discriminative today, which basically tries to determine the probability of a model being true, where we give it certain features So model in this particular case is Y, let's say. What is the probability of a model Y being real if that model has certain features? Features like amino acid sequence, for example, or structural features, whatever. So that's the kind of model that are called discriminative. But very recently, people have come up with computational models that are called generative models, which does the opposite. It determines the probability of a set of features existing if a model is given to you. And some aspect of this new generative models you're probably already familiar, in which people have generated fake faces from completely random people, and these fake faces, fake images of faces, uncannily resemble real people, but no such real people actually exists, but are generated in the computer completely anew. So let's try one of these models, if you like. So let's go to this art maker. I don't know how many of you have been there. So what you do is that you type a text, and you can type whatever you want. And I'm going to type something like, give me a, can you see my desk, by the way? Desktop, can you see? Anyone?
I cannot see your desktop.
Oh, you cannot. Okay, so I have to do a new share. All right, yes, got it. Okay, so this is the desktop I get when I click that link. And this link has a text entry box. So I write, you can type anything you like. So I'm typing, give me a new monoclonal antibody against Ebola pandemic in Uganda. Okay? You can write anything that you want. You can write birds flying through the ocean water in deep blue, algae, forest, whatever. Okay? So I've just written this. Okay? Now, I have to come up with a style, and there are panels of style. And let's see. I mean, this dragon looks pretty interesting, so I'm going to take this dragon and create. Now, what is the computer doing at this stage? The computer has taken this text and has established certain representational learning parameters and is generating from those representational learning parameters, has generated some fake structures and some real structures based on those keywords. And these structures are actually two-dimensional features of images. And look what it has made. This image that you see is generated completely for the first time in this world. Nobody had made this image before. It has made a monoclonal antibody of some type, a picture of that, based on what I wrote. Now, what did I write? Give me a new monoclonal antibody against the Ebola pandemic in Uganda. Of course, this is an art. This is not real science. It's an art. But I'm happy to see that just like an antibody, there are two limbs of these things. So every one of these has two limbs, just like antibodies have two limbs. And some antibodies have, wrapped up protein sequences. So these also seem to have some wrappings around. It seems to be in Uganda. I don't see anything about Ebola, but probably because the computer doesn't even know the word Ebola. So this fun here is just fun of making art. But where is my window? Desktop one. Okay. Just one moment. Let me get back to the share again. Let me stop the share and get back first. I can't find my window. Let me get out of here. Nope. Oh, that's because my slide has gone out. Give me a moment. Yes, can you see my screen again?
I still see your desktop, Dr. Ray. Oh, there you go. Now
How
it
about
works.
now? Yes. Okay. So you've seen that art, and last night I made another art in which I used a different style, and that style was this. Give me some antibodies that neutralize Ebola virus. I didn't ask anything about the features of Uganda, so I didn't get any trees. Uganda has trees, I suppose, some forests, but it has cities, but it ignored the cities. So yesterday I asked for antibodies that neutralize Ebola virus. So it made a representational statement from my text into image, and you see again these two hands, two legs, which resemble the two limbs of an antibody. And it was a different style that I picked, so that's why you see this, this strange city-like environment. But that's fun. That's art. So what you did is make a representational model of your text followed by a generative model of an art. Can we do that for a real antibody? Can we represent the 3D structure of an antigen in some mathematical terms and generate a neutralizing antibody that's going to inactivate that particular antigen? So central to this computational exercise is the so-called generative adversarial network, which is something like a tug of war between versions of the generative model and the random or fake model, the so-called minimax game that I talked about. And as I said, the minimax game is also played in evolution, where the virus is the fittest when it is able to replicate and produce the most progeny, whereas the host is the fittest when the host is able to inactivate the maximum types of viral variants. And it is most successful when it can neutralize the virus by antibodies. And so the virus tries to mutate and generate variants to evade the host's immunity. And the host, in its turn, tries to keep up by generating more different antibody variants. So it's a tug of war that's happening in nature, which we are trying to encapsulate computationally. This is a diagram of how the viral variants have been evolving since its first discovery in Wuhan. For example, right in the at the left end, it says EC. That's where the original virus was identified. That's part of the word December 2019. Only the EC has come out in this picture. And then the virus has evolved through various lineages, just like human lineages of human beings, etc. The viruses have evolved along different lineages. And as you see, as time goes by, the viruses are evolving faster and faster. You see many more lineages appearing over time. Why does that happen? A clue to this may come if you overlay this tree with our vaccination campaign. One dose of vaccine is a lighter shaded diagram, and the number of people who have gotten both doses are in the darker shade. And as you see, there is a near complete correspondence between the frequency with which new variants of the virus, SARS-CoV-2 virus, are arising and the rate at which we have been vaccinating globally people who might be infected, who might accidentally be infected by the virus. And as you know, many people who had been infected in the past by one variant are now being infected by other variant because we do not yet have vaccines for all the variants. So are we generating more variants, which may be less virulent but maybe more infectious, through the vaccination campaign because of this evolutionary tug of war between the evolution of the variants and the evolution of the antibodies through the vaccination? So in order to understand that, we need to learn a little bit about the spike protein, which is on the surface of the virus. And the spike protein has multiple domains, which is on the lower right. This block diagram of the spike protein, and there is a word called RBD, or receptor binding domain, which is the region of the spike protein that binds to the human ACE2 receptor protein in order to internalize the virus. So virus infection is intimately connected with the spike protein's ability to interact with the ACE2 receptor. And if you look at the spike protein, you find that many different mutations have arisen since the Wuhan version came up. So the Wuhan had no other variant. These little Q493R, etc. These are variants, variant amino acids. The first letter is one amino acid, followed by a number, which is the number of the amino acid residue on the protein, followed by another number, which is the new amino acid that has arisen. And this is a compendium of all the variants that were identified until approximately February of this year. If you focus on one region, this pink region, RBD, receptor binding domain, you find that majority of the variations crowd within a small region of this RBD domain. And even within this domain, there is a region which have the most variation, approximately 30 amino acid stretch. And we have identified there are 8 regions that are variable between the original Wuhan version and the Omicron version. And these are diagrammatized here on the right as dots on the RBD domain. Each dot is a variant, and the number on top of it is the amino acid residue number. And W means Wuhan, and O means an Omicron. So if Wuhan had all Ws, and Omicron, let's say BA2, has all O's, then there are 256 possible variants to go from the Wuhan version to the Omicron version, because each position can either have a value of W or a value of O, a Wuhan type or an Omicron type. So if each position has 2 possible sequences, then there are 2 to the power 8 or 256 total variations possible. So these variants are on the spike protein of the virus, and they bind to the human ACE protein with certain affinities, and they also bind to the circulating antibodies that had been raised against the Wuhan version by the vaccine, but the vaccine was not raised against the Omicron version. So the antibodies also have some affinities to this region. So the fitness of the virus is to escape the antibodies and bind tightly to the ACE2 receptor. And the fitness of the host is to find antibodies that bind tightly, more tightly, to the RBD domain so that it blocks the RBD domain's possible binding to the ACE2 site. The question is there an optimum path of fitness through the evolutionary fitness landscape from the Wuhan version to the Omicron version? And you can think of this in these terms. The Wuhan version has a low fitness, and the Omicron version has a high fitness because more antibodies bind to the Wuhan and neutralize it, whereas the Omicron version has fewer antibodies, maybe a single peak, or maybe a rugged peak through many valleys, and people and like people who have to climb mountains, the human body would have to climb fitness peaks by many variations. So what is true? In fact, this is what we are trying to establish in the laboratory by generating these combinatorial variants of the RBD domain, those 256 variants, and then in the laboratory trying to find out their affinities, making computational simulations by docking and molecular dynamics simulations to find how tightly should they bind, and then learning from them by deep learning models the 3D features that allow such binding, and then ultimately do this generative adversarial neural networks to play the minimax games to generate novel antibodies against them. So these are the people who are doing it. Karen is a postdoc in the lab who is leading all the directions of this project. Then Jonathan Felix is working on the variants and molecular determinations in the laboratory. Tristan and Zihao are working on various aspects of the computational part. Jordan was a summer student, and so was Mariana and Kate. They worked on all three aspects. Well, actually, Mariana worked only on the computational aspect and made significant contributions. Jordan and Kate both worked on experimental parts, and they also made very significant contribution during last summer. Both of them are working remotely currently on the project. This is a collaboration with Ilya Tolstorokov, who is a research professor in KGI, Jennifer Hernandez, a professor in the pharmacy department. Stefano Lonardi is a computer science professor in UC Riverside. Again, it's a mistake. I apologize. Okay, I'll correct it later. My apology. He's in UC Riverside, and Matt Suzinski, Pomona College. You can see that I prepared this slides late last night, so there are a lot of typos. Pardon me for that. And here is to end the lab people's photo. We don't just do science, we have some fun as well. That's all.
Thank you so much, Dr. Ray, for all of the information. We really appreciate it. If there's any questions, you can please type them out in the chat or unmute yourself to ask them as well. And also, if there were any other questions as well, please feel free to email me. I'll be putting the email in the chat. If you have any questions about KGI or any questions about our programs or anything to hand over to Dr. Ray, just let me know.
Yeah, I have a question for Dr. Ray.
Of course, thank you.
Could this model be also used to kind of predict like future variations in the spike protein?
Probably not. That's nearly impossible to do.
Because that would be like generating mutations, right?
Correct. Yes. Generating mutants on the basis that which mutations would be selected in evolution. So this spike protein, for example, has about 300 different amino acids. Okay. So there are 20 possible amino acid variation at each of these 300 possible places. Right. The number of variants that can exist is actually quite large in the whole human population, thinking that there are about 8 billion people, 6 to 8 billion people, I forget how many, throughout the earth. And if 90% of them are infected, most probably 90% will be infected by the time this pandemic goes out completely. And within each infected person's blood, there are about 10 to the power 12 different viral particles. You still will not be able to generate as many different possibilities as there are real possibilities. That is 300 positions and 20 variations. So 20 to the power 300 is an astronomical number. So evolution selects for a minor fraction of it for very complex reasons. And these reasons we don't understand. These reasons may have things like survival of the virus within a tiny little particle or a droplet of saliva in the air for more than 2.5 minutes, whereas another variant survives for more than does not survive more than 2 minutes. Okay, So we just don't understand what these properties are that are selected. So it's nearly impossible to predict what variants are going to arise in a pandemic proportion in the future. That we cannot do.
Or another
But
question. Oh, so go ahead, Dustin.
no, I'm just building up. Is that's because we can't determine the environmental factors, or just that there's just too
That is
many?
precisely that. Yeah,
Okay.
just too many as well as too indefinite environmental factors, both inside the human body and outside.
My other question is, are these generated antibodies, would they be immune to like antibody dependent enhancement? You know how for dengue?
Oh, yes,
Okay.
right. So we don't understand antibody dependent enhancement. This has not shown to be a reality so far
Mm.
with COVID 19, most likely. There has not been any definite demonstration of antibody mediated enhancement of the SARS CoV 2 virus. That is not true, however, of several other viruses. For
Right.
example,
Yeah.
dengue virus is one. So mm, it, it's impossible to say that it's not going to happen,
Yeah.
but we still don't understand why antibody dependent enhancement happens in dengue virus. So we don't know.
Thank you.
Any other question? Okay.
All right, it looks like there's no other questions. Thank you so much to everyone for attending today's webinar. We truly appreciate it. And thank you so much, Dr. Ray, for giving us a little bit more insight about the artificial intelligence and defense against emerging pandemics. We really appreciate your time for being here. And I hope you all have a wonderful rest of your day. And again, please let me know if you have any questions. You can always reach out to me or our admissions team. Thank you.
All right. Thank you.